Hi everyone, I hope you all are doing great. So, welcome back to another video. And in this video lecture, we're going to be discussing about a very easy chapter which is called as the enzymes. Now, I know I haven't been uploading very regularly on my YouTube, so extremely sorry for that. I will try to be more regular here. And also, if you're enjoying these lectures, please do make sure to subscribe to my channel and also share this with a lot of your friends so that they can also benefit from these videos. And also, please leave down any of your thoughts, your suggestions in the comment section down below. I would love to read them out. And yeah, let's get started without any delay. So, this is a chapter enzymes. And now, as I said, that enzymes is relatively a very easy chapter, right? So, first of all, we need to know what exactly are enzymes, right? What is an enzyme? Now, the first thing that you need to know that needs to click in your mind instantly, that is that an enzyme is always going to be protein in nature. Okay? An enzyme, it always has to be a protein. So remember that not every protein is an enzyme, but every enzyme is definitely a protein. Okay? So they are proteins in nature. One point, okay? The other point that I want you guys to know is that you have why do we actually need these enzymes? Like, what is the need of an enzyme in our body? Well, you must have heard of the word a catalyst, right? What is the role of a catalyst? A catalyst is something you must have seen in industrial reactions and all, especially in chemistry. We have heard the word catalyst. Now, what does a catalyst do? The main purpose of a catalyst is to speed up your chemical reaction, right? It is there to speed up a chemical reaction so that you can produce a certain type of a product in less amount of time, okay, so that you do not consume a lot of time and you fastly you create your product in less time. So that is what the main purpose of a catalyst does. Now, enzymes in our body work in the same way. Okay, so how do they work? They work as biological catalyst. Okay, they're working as biological catalyst. Why biological? Because they're working inside our body, inside our cells. So that is why they're speeding up certain chemical reactions. They're speeding up your metabolic rates. They're speeding up all your metabolic reactions inside your body. Um, and that is done through the help of enzymes. So they're working as biological catalysts. Now, why do we even need these chemical reactions? Because, guys, let's say if you take a meal, okay, you eat food. Literally, if you do not have these enzymes in your body, that food can take up to 4 weeks, almost a month to digest. And if you have, which we have these enzymes, so that food literally just digests within a few hours. Like within 3 to 4 hours, your food is completely digested. But if you do not have these enzymes, then that same meal, just one meal, is going to be digesting in about 4 weeks. I mean, do you think that we're going to be able to sustain life in that way? No, we are definitely not going to be able to sustain in that way. Life cannot sustain without enzymes. So enzymes are very, very important for us to sustain. Okay, to for life to sustain, enzymes are a crucial, a crucial part of our body. So that is why it is so important to know what enzymes are, to study about enzymes. Okay, now let's exactly see how do enzymes lower our activation energy. Okay, so let me just come to this reaction over here, and the concept that I want to explain over here is that how do enzymes actually speed up your chemical reactions. Okay, so this is what we want to learn, that how do an enzyme, it speeds up your chemical reaction. So let's suppose I have a reactant A and it is breaking up into B and C. It is breaking up into product B and product C over here. Now here, let's say I have about I have about 100 molecules. Okay, I have about how many molecules? I have about 100 molecules of reactant A, and there is this certain barrier. Okay, there is this certain barrier. So let's say this is my ground. Okay, this is my ground, and then there is this certain barrier that each of that if any of these molecules of A, okay, it has to jump over this barrier, and then only they can form this product B and C. Okay, just a hypothetical way of thinking. Okay, I'm trying to explain this concept. I hope you guys get it. So what I'm trying to say is that every molecule over here needs to jump over this barrier right here. Okay, this is the barrier, and we call this barrier as the activation energy. Okay, we call this barrier as the activation energy. What is activation energy? Activation energy is the initial amount of energy that is required to start the reaction. Without this activation energy, my reactant A cannot break into product B and C. Okay, so this is the initial amount, this is the energy that will be consumed by A, okay, to form these products B and C. So let's suppose that the activation energy required over here is 200 joules. Okay, so I have a molecule A. Let's suppose that out of these 100 molecules, one molecule comes, okay, and let's say it has about 200 joules of energy. So it can easily, because this activation energy is also, this is the minimum amount of energy required to start the reaction, and it has about 200 joules of energy, so it easily crosses this barrier. Now comes another molecule from this 100 molecules. Okay, now another molecule comes, and now this molecule, however, let's say has 150 joules of energy. Do you guys think that this 150 joules of molecule, um, this 150 joules of energy molecule is going to cross this activation energy barrier? No, it cannot cross this energy barrier. Why can it not cross this energy barrier? Well, it cannot cross this energy barrier because it has energy. It has about 150 joules of energy, which is less than the activation energy. So this molecule cannot cross this activation energy ka barrier. Okay. Now let's say another molecule comes and it has about 250 joules. Then it can definitely. So let's say that around 30 molecules from here, okay, around how many, around, let's say around roughly, out of these 100 molecules, only 30 molecules are such that have energy that is either equal to 200 joules or more than 200 joules, so they're easily able to cross this barrier and they're able to form the products B and C. But those other 70 molecules, I literally have 70 molecules that are unreacted. Okay, 70 unreacted molecules of air, they're not able to form the product B and C. So here my yield, okay, my products are going to be very less. But if I introduce a catalyst over here, okay, if I introduce a catalyst over here, what is going to happen is that this catalyst is going to be, what is it's going to do is that it's going to lower my activation energy. Okay, it literally lowers this barrier over here, that this barrier that you guys see over here. What it does is that initially it was at this height, right? What it does is that it lowers this activation energy literally from this point, it lowered it to this point. So now those molecules, around 70 molecules, which had energy less than 200 joules, okay, varying energy. Okay, so let's suppose this catalyst brought the lowered my activation energy to around 50 joules. So now even the molecules, the 70 unreacted molecules which had energy less than 100 joules, okay, and roughly around 50 joules, those molecules can now jump over this activation barrier. They have this minimum amount of energy to gain this energy and break into product B and C. So hence that literally says that that is going to increase my products, because now more molecules of A react and more products are going to form. So if more products are going to form, then ultimately I can say that my rate has increased. So do you guys see the concept over here? How literally enzymes work to lower your activation energy. I hope you guys get the concept of activation energy. It is very important. Okay. So far we have discussed upon the properties of enzymes. Okay. So what are enzymes? They're protein in nature. Okay. This is the first important thing that you need to know, that enzymes are proteins in nature. The second point is that they're acting as biological catalyst. So proteins, biological catalyst, and they also speed up your chemical reaction. How do they speed up your chemical reaction? By lowering your activation energy, and they are extremely important to sustain your life, because if you do not have these, if you do not have these enzymes, life would not be, we would not be able to, we would not be able to sustain. Okay, we would not be able to live up. Okay. So this is primarily what you guys need to know about enzymes. Now let's see that how do exactly enzymes work. Okay, how do an enzyme work? So first thing you need to know, if I just take out a structure of an enzyme. Okay, so let's suppose this is a certain enzyme. Okay, this is an enzyme, and just a second, let me just make this a little smaller so you guys actually understand. Okay, so let's suppose that I have this enzyme. Okay, this is an enzyme. Always remember that an enzyme will have a 3D structure. Okay, so an enzyme, it has a 3D shape. It is not a 2D molecule, it is not a 1D molecule, it is a 3D shape. Okay, it has a three-dimensional shape, and some every enzyme is going to be having a place called as the active site. So this portion that you see over here, this mouth type thing that you see over here, this is going to be an active site. And we say that enzymes are super specific in their reaction. Okay, enzymes are extremely specific. Why? Because for every specific reactant, okay, and I can call my reactant as the substrate. Substrate is the one on which my enzyme, substrate is basically the molecule on which my enzyme is going to act on. Okay, for example, the reactant. Okay, if it had to break that reactant, so I'm going to call that reactant as my substrate. Okay, I'm going to call that as a substrate, and my substrate and my enzyme are going to be complementary to one another. They're going to be complementary to one another. What does this mean? It simply means that if this was my enzyme, so this is going to be my substrate. Okay, so this red color thing over here, this is something. So you see this shape and this shape of the substrate, the shape of the active site. Okay, the shape of the active site is complementary to the shape of this substrate. So that means that for every substrate, guys, for every substrate, my enzyme is going to be specific. So not every enzyme works on every substrate. Okay, not every enzyme works. So that's why enzymes are highly specific. They cannot break or make, they cannot work on all types of substrate. There are specific enzymes for specific substrates. Okay, so for example, let's come over here. Okay, let's come over here. So here you see that we have this in yellow color, we have this substrate. Okay, so this is my substrate. Let me So this is my substrate, this yellow color. Okay, this substrate. And as you can see, notice the shape of the substrate. Okay, it's something like this. And then notice the shape of this active site of the enzyme. This is the active site. Okay, this is the active site. This is the part where my substrate during the reaction is going to be binding to my enzyme. Okay, for the reaction to take place. And this, you can see that this substrate is specific to enzyme 1 only. Okay, this is enzyme 1, and it is complementary, it is complementary to the shape of this substrate. But notice over here this enzyme too. I have a second enzyme as well. But this enzyme cannot act on this substrate. Why? Because the active site of this enzyme too is not complementary to my substrate. I hope you guys understand the specificity of enzyme and substrate, okay? So, important point. And we call it as the enzyme specificity is defined, like it is defined by the model of lock and key, okay? So, enzyme specificity, okay, like who actually, what model explains that, how enzymes are specific, that is actually described by the lock and key model. Now what does lock and key model states? Let us understand that, okay. So lock and key model basically tells us that you guys have seen a lock, right? And this is how a lock is. And inside this lock, you insert a key over here. Okay, so you insert a key and then this opens up. Your lock opens up. Similarly, your lock is your enzyme. Okay, so this is your enzyme. Your lock is your enzyme. Okay, so your enzyme is acting as your lock, and into this enzyme a specific substrate binds to it, just like how a key enters into a lock. Similarly, a substrate acts as your key and it enters into your enzyme acting as the lock. I hope this is clear to you all. Now when an enzyme it binds to a substrate, okay, so it forms something called as an enzyme substrate complex, okay. So let me explain how exactly this happens. All right guys, so here what I want to explain over here is that, notice over here I have an enzyme over here, okay. So this red color thing is going to be called as my enzyme. Okay, just a second, let me just change the color. Yeah, that's it. Okay, so this is my enzyme. Okay, so this is my enzyme, and then this is my substrate. So that's my enzyme and that's my substrate. So in this reaction, my substrate is A. Okay, so this is my substrate A, the reactant on which my enzyme is going to be working. Now this substrate joins, because they were complementary to one another, the enzyme and the substrate, they were both complementary to one another. So they form something that is called as the enzyme substrate complex. Okay, they form something that is called as the enzyme substrate complex. I'm not going to write the whole thing out, but yeah, just get the point. They're going to be called as enzyme. अच्छा, चलो लिख लेते हैं, enzyme substrate. Okay, so because this is your enzyme and this is your substrate, they're both binding temporarily over here. Okay, it's not a permanent they're not permanently joining it to one another. So this was initially my enzyme and this was my substrate. They both joined together, they formed some. This is according to the lock and key model, okay. This is what your lock and key model explains about enzyme specificity, that there's an enzyme, there's a substrate. The active site of your enzyme is complementary to the shape of the substrate, okay. Every active site is specific to only one type of a substrate. Not every active site is going to be working on all types of substrate. We have already discussed this, okay. And then it forms something called as the enzyme substrate complex, and then that enzyme substrate complex forms. So let's over here that the work of this enzyme was to break this reactant, my substrate, into two products. What were those products? B and C, right? So it has broken them up over here. The active site has worked on them, and then this is something that we call as the enzyme product complex, because this is your enzyme and then these two are your products, but they've not been released yet from the enzyme. So we call this as the enzyme product complex. Okay, we call this as the enzyme product complex. And then finally, at the end of the reaction, my enzyme leaves the two products and I'm left with B and C. And always remember one thing, that your enzyme, it remains unchanged at the end of a reaction. Okay, your enzyme, just like we call it as a catalyst, we're literally calling it a biological catalyst. So just like a catalyst is unchanged at the end of the reaction, similarly my enzyme is also unchanged at the end of the reaction, and this can be used multiple times. Okay, the same enzyme can be used to break multiple same substrates. Okay, so the same enzyme can be used over and over again, so it is reusable. Okay, this is also an important point to know over here. Unless and until there are some factors that affect enzymes, and that is something that we're going to be discussing about in the next video, that what are some factors that affect your enzyme activity. Otherwise, your enzyme remains unchanged at the end of a reaction, and it is reused. Okay, it can be reused for the same substrate again and again to continue a reaction. This is something that we have talked about. So again, this is something I want to talk about again. This is the same reaction that I just explained over here. That these are your these are your substrates, and this is your enzyme, and then they form something. When the substrate joins to your enzyme, it forms something called as the enzyme-substrate complex, and then before the products are actually released, they form something called as the enzyme-product complex, and then this is the products that have been released over here. So far, this is for this video. I hope you guys enjoyed it. You learned something new from this video, and stay tuned for the next video, because in the next video, as I already said, we're going to be explaining about the factors affecting the enzyme activity, and then our chapter is wrapped up. Okay, so yeah, that's it. Bye everyone.